Welcome to Base by Base, the papercast that brings genomics to you wherever you are. Thanks for listening, and don't forget to follow and rate us in your podcast app. So, I want you to think about the last time you caught a truly awful virus.
Oh man, yeah, we all have that one memory.
Right. And if you're like most people, your mind immediately goes to the symptoms. You remember the fever, the bone-deep body aches, the chills.
Just being completely stuck in bed for days, feeling entirely drained.
Exactly. We naturally tend to view viruses as these violent, short-term invaders. Once the fever breaks and your energy finally comes back, you basically consider the battle won.
The invader is cleared, your immune system did its job, and case closed.
Case closed. But what if the battle isn't actually over? What if, while you were distracted by all those acute symptoms, that virus was quietly leaving behind an invisible, permanent molecular scar deep inside your DNA?
A scar that, might not actually cause a problem until years, maybe even decades later.
And how could this change everything we know about long-term health? That invisible wreckage is exactly what we are exploring in today's deep dive. Today, we celebrate the work of Erica Lamkina and her team, who have advanced our understanding of exactly this hidden viral aftermath.
Yeah, we're looking at a really fascinating open access paper from PNAS published in 2026.
And our mission today is to unpack the incredibly stealthy ways the dengue virus, specifically a somewhat understudied serotype called DENV-4, completely sabotages our cellular repair mechanisms.
We're going to explore how this virus manages to keep our cells alive just long enough to use them as a manufacturing plant, all while completely trashing the genetic blueprint inside.
It is a remarkable piece of research because it directly challenges long-held assumptions in virology, right?
Yeah.
I mean, dengue is a massive global health issue.
It's huge. We're talking about up to 400 million infections a year globally.
Wow, 400 million.
Yeah, and due to climate shifts, we are seeing diagnoses increasing in places like the southern United States, which is a major shift.
But despite those staggering numbers, the underlying molecular mechanisms like how the virus actually interacts with and manipulates our host DNA over a prolonged period have remained largely a mystery.
Right, which is why this paper is so crucial. It finally pulls back the curtain on that specific mechanism.
Okay, let's unpack this. To really understand how DNV-4 damages the body, we first have to look at how it enters the scene of the crime, so to speak, without setting off the immediate alarms.
Right. The researchers set up a very deliberate experiment here. They took host cells, specifically monkey kidney cells and human liver cells.
Which are VeroE6 and HUH7.
Right. Exactly. Those are the standard models for this kind of work. And they infected them with DNV-4, but they did it at a very low dose.
A multiplicity of infection, or MOI, of just 0.1, I believe.
Yes. And they watched them over a 5-day period. That low dosage is a critical design choice.
Why is that so important?
Well, it basically means that initially only about 1 in 10 cells is actually infected. They aren't overwhelming the cell culture with an artificial tsunami of the virus.
Which would just cause immediate toxic shock, right? The cells would just die instantly.
Right, exactly. Instead, they're simulating a natural creeping infection. This allows the virus to slowly replicate and spread over those 5 days.
So they wanted to monitor the prolonged viral dynamics and see how the host cell adapts over time, not just how it dies under a massive viral load.
Exactly. And the really surprising part of this creeping infection is that the cells didn't just immediately die off.
Yeah, if you looked at them under a microscope, they looked completely fine.
Totally fine. The researchers checked their viability, and it was normal. They even looked at a whole dashboard of genes responsible for keeping the cell dividing and functioning.
The engines of basic cellular life. Yeah. Genes like MCM7, ORC1, POLY1, and PRIME1.
Right. The transcription of all those core proliferation genes stayed totally stable. The cells were just humming along, going about their business.
It's so deceptive. Like, I was thinking about this, and it's basically like a burglar breaking into your house. But instead of smashing the windows, they change the locks, brew a pot of coffee, and set up a massive counterfeiting operation in your living room.
That's a great way to put it.
Right. While keeping the house looking perfectly intact from the outside.
Yeah, that lack of immediate physical damage is what we call a minimal cytopathic effect. The cells are physically intact, their basic operational machinery is running.
Which is pretty clever of the virus.
From an evolutionary standpoint, it's a highly evolved strategy. You have to remember, the virus is an obligate parasite. It has absolutely no machinery of its own.
So it relies entirely on the host cell to act as its factory to print more viral RNA.
Exactly. If the virus just immediately triggers cell death, the factory shuts down and the virus can't reproduce. The virus needs the cell alive.
But as we see in this study, Keeping the cell alive is not the same thing as keeping it healthy.
Not at all.
Here's where it gets really interesting. If the outside of the cell looks totally fine, and those basic proliferation engines, the MCM7 and ORC1, are still running, how did the researchers actually figure out that the foundation was rotting?
They looked for a very specific molecular distress signal called gamma H2AX.
Gamma H2AX.
Yeah, you can think of gamma H2AX as a chemical flare that gets sent up the moment a cell experiences a double-strand DNA break.
Okay, let's visualize that.
Yeah.
A double-strand break isn't just a minor typo in the genetic code, right? Like, if DNA is a suspension bridge, we aren't talking about a loose bolt on the walkway.
No, not at all.
We're talking about one of the main steel support cables completely snapping in half.
That is the exact level of severity. It is the most toxic, catastrophic form of DNA damage a cell can endure.
Wow.
Yeah, if a snapped main cable isn't fixed, the entire structure becomes unstable. In a cell, if those breaks aren't repaired, the cell will either become highly cancerous or will recognize the catastrophic failure and trigger apoptosis.
Which is programmed cell death.
Right. So the researchers monitored this gamma H2AX chemical flare over the 5 days. At 24 hours, they saw a baseline level. At 48 hours, the flares start going up.
And by day 5?
By day 5 of the infection, the levels of gamma H2X were up to 24-fold higher than normal in the surviving cells.
24 times the normal amount of catastrophic DNA damage. And yet the cell is still just sitting there looking fine on the outside.
It's wild. The virus, simply by operating its factory, is heavily and systematically fracturing the host cell's structural DNA.
But normally, if a main suspension cable snaps, the bridge authority doesn't just ignore it. They shut down traffic and bring in a massive structural engineering team to fix it. Why wasn't the cell doing that?
Under normal circumstances, that is exactly what happens. The chemical flare triggers what's called the DNA damage response, or DDR.
The cellular repair crew.
Exactly. The DDR is this incredibly complex rapid response network. It pauses the cell cycle, shutting down the traffic, to use your analogy, and recruits specialized repair proteins to meticulously rebuild that broken cable.
But they didn't show up here.
No. The researchers ran a massive genetic audit using a 96-well gene panel. They were looking at the expression of the genes responsible for this repair crew, and they found a huge plot twist.
The repair crew wasn't just failing to show up, right? Their blueprints had been completely suppressed.
Yes. The virus is actively blocking the cell from manufacturing the repair crews. It suppressed the transcription of almost all the major repair pathways.
We're talking about pathways like BER, HR, NHEJ, and NER.
Well, all of them. BER, or base excision repair, which fixes small chemical changes. HR and NHEJ, the heavy-duty crews that rebuild those double-strand breaks. And NER, the teams that cut out UV damage.
And the genes instructing the cell to build those proteins were shockingly silenced.
Silenced. And what's fascinating here is that it goes even higher up the chain of command. The researchers found that the virus suppresses a master regulatory protein called ATR.
Total ATR protein was down. That's like the chief structural inspector.
Exactly. ATR's job is to survey the damage, sound the general alarm, and coordinate the entire response. By day 5, the production of that chief inspector was actively shut down.
So it is a dual-pronged attack. The virus is running its factory so recklessly that it causes massive structural damage to the DNA, and simultaneously, it intercepts the communications to the chief inspector, so the repair crews are never even built.
Precisely. It is intentionally dismantling the cell's ability to fix its own blueprint. This ensures the viral replication can proceed without the host cell pausing its cycle to make repairs.
Wait, but if the virus is trying to stop the cell from repairing itself, wouldn't it just shoot itself in the foot? I mean, if the structural damage gets that extreme and there is absolutely no maintenance happening, the cell's internal sensors would eventually trip.
Right, the cell would undergo apoptosis.
And the viral factory would close anyway. There has to be a catch. Were there any exceptions? Did the virus really shut down every single repair pathway?
That is the exact paradox the researchers investigated, and it reveals how highly selective DENV-4 actually is. It doesn't indiscriminately cut all the phone lines.
Okay.
They found one major exception. A specific pathway called mismatch repair, or MMR, remained completely unchanged.
Mismatch repair. So proteins like MLH1, MSH2, and MSH6.
Yes, the transcription of those key MMR proteins was totally stable despite everything else being suppressed.
Why leave just the mismatch repair crew working while the rest of the factory's maintenance is shut down?
The researchers hypothesized this is a very calculated survival tactic. As you pointed out, if the genome fractures entirely, the cell self-destructs. The virus doesn't want that.
Right.
Mismatch repair is generally responsible for fixing small mispairings that happen during normal DNA replication. By keeping just this one pathway functional, the virus might be allowing the bare minimum baseline stability required to keep the cell alive and the factory running.
It's like doing just enough basic maintenance to keep the building from collapsing today, even though the foundation is fundamentally compromised for the long term.
Exactly. It's doing the bare minimum.
That is incredibly manipulative, and what strikes me is how specific this strategy is, especially when you compare DENV-4 to other major viruses we've studied recently, because they don't all use this same playbook, do they?
Not at all. And that comparative biology is where this study gets really illuminating. If we look at other prominent RNA viruses, their strategies are entirely different.
Innocence.
Well, both influenza and SARS-CoV-2 actively and aggressively disrupt that exact same mismatch repair pathway that DENV-4 intentionally leaves alone.
Wow, so they target the exact thing DENV-4 protects. And doesn't SARS-CoV-2 also go after the telomeres?
Yes, it famously damages them.
Right, and just as a reminder, telomeres are the protective caps at the ends of our DNA strands. Think of them like the thick plastic coating at the end of a high-tension wire. If you strip that coating off, the internal wires fray, the DNA unravels, and the cell ages and dies very quickly.
That's a perfect way to picture it. SARS-CoV-2 heavily damages that protective coating. But when the researchers tested the telomeres in these DENV-4 infected cells, they found no damage.
And not at all.
None. They measured the actual physical length of the telomeres, and they measured the TRF2 protein, which is responsible for maintaining that cap. The result was totally stable. DENV-4 leaves the protective caps completely alone.
So SARS-CoV-2 strips the protective wire coating and breaks the mismatch repair crew. DENV-4 leaves the coating alone, leaves the mismatch repair crew alone, but completely destroys the main cables and gags the chief inspector.
Right.
It's running a completely different software program to hack the cell. But from the paper, it seems DENV-4 doesn't just act differently from respiratory viruses, it acts completely differently from its own dengue siblings.
This raises an important question, actually. How much do we really know about the dengue virus family? Because they are made up of 4 main serotypes, right? DENV1 through 4.
And they share about 65% of their genetic code. Clinically, if you get infected by any of them, your symptoms look very similar.
Exactly. But at the molecular level, they behave like entirely different pathogens. For example, the researchers look at a cellular process called autophagy,
which is the cell's internal recycling system. It's a stress response where the cell gobbles up its own damaged components to clear out the trash and survive.
Yes. And previous studies have shown that dengue serotype 2 actively ramps up this recycling system. It uses autophagy to help itself replicate. DENV2 thrives on it.
But what about DENV-4?
Well, when this team looked at specific protein markers for autophagy, specifically LC3IN2 in DENV4, they found the exact opposite.
The exact opposite.
DENV4 actually decreased those autophagy markers. It suppresses the recycling system entirely.
Why would 2 viruses that are 65% identical evolve completely opposite strategies for hijacking a cell?
It speaks to evolutionary niches, really. Over time, these serotypes likely evolved in slightly different host populations or mosquito vectors, forcing them to develop unique pathways to achieve the same ultimate goal, replication.
So DENV2 might need the recycled materials from autophagy to build its viral particles, while DENV4 might find the autophagy process threatening to its specific replication strategy, so it shuts it down.
Exactly.
That has massive implications for how we treat this, doesn't it? We generally talk about dengue as one monolithic disease. But if serotype 2 and serotype 4 are fundamentally different at the molecular level, how do you even begin to design a universal antiviral drug?
It highlights a massive, glaring gap in our current medical approach. We cannot study one serotype in a lab and assume the others will respond to the same therapeutic intervention.
Right.
If you design a drug that targets the autophagy pathway based on DENV2 research, it might be completely useless or even counterproductive against DENV4.
Wow.
This is a huge hurdle for developing pan-dengue vaccines or universal treatments. We have to understand the specific host cell pathologies of each serotype.
So what does this all mean for the listener? Let's pivot from the cellular mechanics to the human impact. Because it's easy to get lost in the brilliant, sneaky ways this virus operates. But the ultimate question for you, the listener, is, what happens to a human being walking around with these surviving genetically fractured cells in their body?
Right, because the fever is gone, the acute infection is cleared.
But these cells survived with 24-fold higher DNA damage and no primary repair crews. How does a cell with broken DNA actually patch itself up enough to keep functioning?
It gets desperate, and this is where the researchers utilize quantitative PCR, or qPCR.
That's a highly sensitive technique used to measure exactly how much of a specific gene's instructions are being pumped out by the cell, right?
Exactly. And while the normal, highly accurate repair blueprints were suppressed, the qPCR data revealed that a very specific alternative gene called P-iota was highly upregulated.
P-iota, what is its job in this desperate scenario?
P-iota is a highly mutagenic polymerase involved in a backup system called translesion synthesis. Normally, when the standard cellular machinery is copying DNA and hits a giant pothole of structural damage, it stalls out. It waits for the DDR crews to come fix the road.
But because DENV-4 suppressed those crews, the cell tags in P-iota.
Right. Think of P-iota as a sketchy, rapid response backup crew. It doesn't meticulously read the genetic code to rebuild the sequence accurately.
It just looks at the broken gap and aggressively shoves random genetic letters in there to bridge the hole, just so the replication process can keep moving.
Yes, and because of that, it is highly error-prone and extremely mutagenic.
Which is an incredibly sloppy patch job. It's essentially paving over the pothole with whatever trash is lying around just to keep the road physically connected. It's literally guessing the genetic code.
It is guessing. And by forcing the cell into a state where it has to rely on P-iota, the virus isn't just leaving damage behind. It is forcing the host cell to actively cement random mutations into its own permanent genetic code.
The researchers refer to these in the paper as durable biological scars. And that phrasing is chilling. I mean, the acute fever, the chills, the body aches of dengue, those might be gone in a week. But these randomly guessed genetic letters, these cemented mutations, they are permanent.
They are written into the cellular blueprint for the rest of that cell's life, and they get passed down to every subsequent cell it divides into.
That's terrifying.
And if we connect this to the bigger picture, this molecular reality perfectly contextualizes some very concerning epidemiological data that has emerged recently.
Right. There was a major population cohort study, I think published in 2020, that linked a history of dengue infection to a significantly increased risk of developing leukemia.
Yes. But the crucial detail is that the strongest association doesn't happen immediately during or right after the infection. It occurs several years down the line.
Because it takes years for those subtle random mutations cemented by P-iota to accumulate, compound, and eventually push that lineage of cells into becoming cancerous.
Exactly. The authors of this paper argue that the DENV-4 infection creates a disease vulnerability window.
A vulnerability window.
Yeah, the viral infection is cleared by the immune system, but that brief period where the DNA was heavily damaged, the accurate repair was suppressed, and the mutagenic backup systems were active, that period leaves a lasting legacy.
It incrementally, permanently increases your baseline risk for chronic diseases, including post-Dang syndromes and carcinogenesis, the formation of cancer, years after you thought you were fully recovered. That is a staggering paradigm shift for how we view infectious diseases. And we've spent this deep dive unpacking the stealthy, brilliant ways this virus operates. DENV-4 creeps into your cells, keeps the lights on, keeps the basic proliferation genes like MCM7 humming, and completely bypasses the alarms.
Yep.
Then, while the cell looks physically fine on the outside, it shatters the DNA blueprint. It intercepts the communications to the Chief Structural Inspector ATR, so the main repair crews are never built.
And finally, it forces the cell to rely on a sloppy, error-prone backup system to survive, cementing permanent mutations into the genome.
The cell survives the virus, but it survives fundamentally altered. It becomes a carrier of error-riddled biological scars that can quietly haunt the host's body long after the immune system has claimed victory.
Which is the central insight here. Survival is not the same as recovery.
It really makes you look at a simple passing fever in a completely different light. And I think it leaves you with a rather profound, slightly haunting question to ponder on your own. If a fever you suffered through for a single week in your 20s can leave behind a lifetime of altered, scarred DNA hiding in your cells.
Wait, let me rephrase that. If a fever you had for a week can leave behind these durable scars, how many of our seemingly random chronic diseases or sudden cancers that appear to come out of nowhere later in life are actually just the quiet, delayed echoes of a virus we thought we successfully fought off decades ago?
It's definitely something that changes how you view your own health history. Absolutely. This episode was based on an open access article under the CC BY 4.0 license. You can find a direct link to the paper and the license in our episode description. If you enjoyed this, follow or subscribe in your podcast app and leave a 5-star rating. If you'd like to support our work, use the donation link in the description. Now stay with us for an original track created especially for this episode and inspired by the article you've just heard about. Thanks for listening, and join us next time as we explore more science, base by base.
System sags, warnings flare in a broken chain, but fix-it crew don't show the same.